we have molecules targeting the replication of the virus. For example, one is called remdesivir, and it blocks the way the virus copies its genome. Another antiviral that we have is called Paxlovid, and instead blocks the protease, that is another protein of the virus that separates pieces of proteins. Then we have another class of drugs that it's not an antiviral per se, but they work against the virus, and these are monoclonal antibodies. So monoclonal antibodies are mimicking the response of the human body to the virus, so the rays of the immune system, but these antibodies are produced in vitro. And we have several monoclonal antibodies that show the efficacy against SARS-CoV-2, but some of them already failed because of new variants of SARS-CoV-2 arising, but some are still active. One of the limits of antiviral drugs is the resistance, because the virus can easily, but some easier than others, develop resistance to a drug. Other problems related to the antivirals are the fact that when a new virus emerges, we don't have any antiviral available normally, because normally the antivirals are specific for each virus. So what we lack is a broad spectrum antiviral. Instead, for example, when we have a bacterial infection, we don't have to know which is the bacteria that is infecting us, and we just take a broad spectrum antibiotic. This doesn't exist for viruses. COVID-19 pandemics taught us that we need to be prepared with something that can be broad spectrum. So many labs are working on strategies potentially broad spectrum, or at least on molecules that can be adapted quickly to emerging viruses.